साझा टीवी देख रहे साढ़े सारे ही प्यारे ਸਰੋਤਿਆਂ ਨੂੰ ਮੇਰੇ यानी कि सपनीत कौर गिल वनियो प्यार भरी सत श्री अकाल। इस वक्त पहुंचे हुए जी पैसिफिक एग्रीकल्चर शो 2026 डे 3 ਦੇ ਵਿੱਚ ਜਿੱਥੇ ਕਿ BC ਬਲੂਬੈਰੀ ਕਾਉਂਸਲ ਵनियो ਇੱਕ ਬੂਥ ਲਾਇਆ ਗਿਆ। ਅਸੀਂ ਤੁਹਾਨੂੰ ਪਹਿਲੇ ਦਿਨ ਦੀ ਦੂਜੇ ਦਿਨ ਦੀ ਕਵਰੇਜ ਦਿਖਾਈ ਆਪਣੇ ਟੀਵੀ ਚੈਨਲ ਤੇ। ਇਸ ਦੇ ਨਾਲ ਨਾਲ ਅੱਜ ਜਿਹੜੇ ਤੀਜੇ ਦਿਨ ਦੀ ਕਵਰੇਜ ਹੈ ਉਹ ਵੀ ਤੁਹਾਡੇ ਨਾਲ ਸਾਂਝੀ ਕਰਨ ਜਾ ਰਹੇ ਹਾਂ ਤੇ ਇਸ ਦੇ ਨਾਲ ਨਾਲ BC ਬਲੂਬੈਰੀ ਕਾਉਂਸਲ ਦੀ ਸਾਰੀ ਟੀਮ ਦੇ ਨਾਲ ਕਾਉਂਸਲ ਮੈਂਬਰਸ ਦੇ ਨਾਲ ਗੱਲਬਾਤ ਕਰਾਂਗੇ। ਜਿਹੜੀਆਂ ਜਾਣਕਾਰੀਆਂ ਉਹ ਸਾਂਝੀਆਂ ਕਰਨਗੇ ਉਹ ਤੁਹਾਡੇ ਸਾਰਿਆਂ ਤੱਕ ਪਹੁੰਚਾਉਣ ਦੀ ਕੋਸ਼ਿਸ਼ ਕਰਾਂਗੇ। ਇਸ ਦੇ ਨਾਲ ਨਾਲ ਸੁਦੇਸ਼ਨਾ ਵनियो ਇੱਕ ਖਾਸ ਗੱਲਬਾਤ ਕੀਤੀ ਜਾਊਗੀ। ਸੋ ਅਸੀਂ ਸਾਰੀ BC ਬਲੂਬੈਰੀ ਕਾਉਂਸਲ ਦੀ ਟੀਮ ਦੇ ਨਾਲ ਗੱਲਬਾਤ ਕਰਕੇ ਤੁਹਾਡੇ ਤੱਕ ਜਾਣਕਾਰੀਆਂ ਸਾਂਝੀਆਂ ਕਰਦੇ ਹਾਂ। ਚਲੋ
Our next presentation is titled Blueberry Virus Testing Update. Please welcome Dr. Peter Ellis, the Laboratory Director at Phytodiagnostic Company, who will be joining us remotely to share virus testing result from 2025 session. And I will be helping him with the questions. If there is any questions, please raise your hand and Mike will follow. Thank you.
Thank you very much for inviting me to your conference. We're here on sunny and cool Vancouver Island. I'd first like to introduce my laboratory crew that work on blueberry viruses. Gerda de Villiers does all the PCR testing in my laboratory. Megan does the sample preparation for blueberries and the ELISA testing, and I'm involved with both PCR and the ELISA testing. A brief short summary. Bob Martin found blueberry scorch first in BC back in 2000. It's spread by aphids and infected planting stock. All high bush blueberry cultivars are susceptible, and the severity depends on the strain of blueberry scorch and the cultivar. Blueberry plants don't recover from blueberry scorch following infection, in contrast to blueberry shock. And infected blueberry shock plants behave as a virus reservoir for continued spread within the field. Transmission occurs by aphids, usually between May and August in the Fraser Valley and in Western Washington. It's relatively inefficient. Earlier work with aphid transmission shows that 10 to 15% transmission when you're using groups of 25 aphids. This is quite different than what we'd see, say, in potato viruses with PVY, where green peach aphid is about 90% effective with single aphids. So it's fortunate that it's relatively inefficient. There's a 1 to 2 year latent period, maybe longer in some cultivars following infection, and it's not clear how much transmission occurs during the latent period. I'm going to review the results from 2020 to current to 2026. These are samples submitted by the BC Blueberry Commission submissions. In 2020, we tested 94 farms, 1,676 samples. As you can see from the table, blueberry scorch single infections were found in 46% of these submissions, and co-infections with scorch and shock was in 6. So that would 55% of the plants we tested were tested positive for shock. Blueberry scorch was blueberry shock was much less at 12%, plus the 6% co-infection would give you about 18%. Roughly a third of the plants tested negative for both viruses. We had much better submissions in 2021, where 186 farms submitted samples, over 4,000 samples. Again, 45% of the samples tested positive for blueberry scorch, and another 5% with a co-infection with blueberry scorch and shock. So we're seeing half the samples testing positive for blueberry scorch. And with the combined single infection and co-infection with blueberry shock, roughly 26%, and a little less than a third of the samples testing negative for both viruses. 2022, we had a lot of samples come in from 234 farms, over 5,000 samples. Again, we're seeing the same thing. If you look at scorch, both single infections and co-infections with shock you're looking at 55% of the samples testing positive. I mean, that's of all these samples that come in. Again, a third of the samples test negative for both viruses. In 2023, we saw something unusual. It was the first year we saw more shock samples than we did blueberry scorch samples. So something happened during the year that suppressed the scorch symptoms, because the samples are usually are collected on the basis of symptomology. So for some reason in 2023, the plants infected with scorch showed less symptoms. And even with that, we're still seeing over 30% of the samples coming in, both single and co-infections, positive for blueberry scorch. And again, roughly a third of the samples negative for both viruses. In 2024, we saw a more typical year. Again, over 50% of the samples testing positive. Fewer farms, 89 farms, and about a little over 2,000 samples came in. Again, we're seeing about a third of the samples negative for both viruses. 2025, we saw many more of the samples, roughly the same number of farms, one more farm, 90 farms with just under 3,000 samples come in. Blueberry scorch, 50% single infections, 7% co-infections, for basically 64% of the samples positive for blueberry scorch. Part of this may be due to the fact that we're changing the format of the ELISA test to make it more sensitive, because the concern is on the samples that test negative for both viruses. You can see the negatives for both viruses drop from about a third to about 24%. Again, ELISA is not as sensitive as qPCR, but ELISA is much cheaper to use for mass testing. So in summary, for the 6 years from 2020 to 2025, we tested over 20,000 samples. And as you can see from the combined results, over 50% of the samples test positive for blueberry scorch, both as singles and co-infection with blueberry shock. And you're looking at about 24% of the samples testing positive for shock. Negative for both viruses are just under about a third of the total samples submitted. Now, these are plants from blueberry farms. If you look at the plants we test for export, we've been testing plants for planting for export to the US since 2008. And we test more than 10 times the number of samples going export as we test for blueberry farms in BC. As you can see, for the combined numbers at the bottom of this table, we tested over 200,000 plants. And if you look at the blueberry scorch and blueberry shock positives, very, very few of the plants test positive. In 2021 and 2022, we saw a few plants go positive. These were new growers trying to get into the export market. Unfortunately, some of these growers were too close to a source of inoculum and had some infections in their propagation material. In 2025, we had one plant that tested positive. It wasn't blueberry, it was red huckleberry, Vaccinium parvifolium. So plants, other Vaccinium species as well as blueberry are tested for export. So what's going on with the blueberry scorch and shock negative symptomatic samples? Some of the samples we receive in the lab, not all of them are collected by specialists. People sometimes send us in dead tissue. We do our best to recover what we can from this material, but dead tissue rarely ever leads to positive samples. Samples sometimes arrive in very poor condition. When people collect samples in the field, there's field heat in the sample. It goes into a plastic bag. If there's any delay in the samples being shipped to the lab, sometimes they degrade quite a bit. And in fact, sometimes if there's a delay in shipping, as the samples come into the lab, they have a rare jam-like smell to them. We do our best to test them to see what we can find, but sometimes the samples are just in too poor condition. I think the main problem is that virus titer may be too low in sample tissue to be detected by ELISA, or it may be unevenly distributed within the plant. And uneven distribution of viruses in blueberries is not uncommon. Uneven distribution occurs with scorch, it occurs with shock, and it occurs with tobacco streak and other ILAR virus that occurs in eastern North America. And it also occurs with another blueberry virus called blueberry red ring spot. So it's not uncommon for blueberry viruses to be unevenly distributed in the plant. If a consultant gets a hold of us and says, look, these plants are clearly sick, they look like scorch or shock, what I recommend is they send us 10 to 20 individual leaves collected from all over the plant and to send them in and we'll test them as single leaves. When we have done this, in cases where we've got 20 leaves, sometimes we'll see 1 to 3 of the leaves test positive and the rest negative. And we see that with both scorch and shock. As I say, if they send us 10 leaves, sometimes we'll see only 1 or 2 of these leaves test positive. So uneven distribution is not our friend. Symptoms from other causes, including insect feeding, frost, chemical injury, or other environmental problems. Unknown viruses or new strains or mixed infections are another possibility. In the last few years, there's been 3 different ludioviruses reported in blueberries. None of these ludioviruses have been described as causing symptoms in blueberries, but maybe when they're mixed with other common viruses like blueberry latent virus and blueberry mosaic-associated virus, these viruses aren't uncommon, and we're not routinely testing for them. Occasionally we do, but we're not routinely testing for these for the samples submitted by the BC Blueberry Commission or Council. Again, other diseases like mummyberry or bacterial canker can confuse people when they're collecting samples that basically aren't virus problems at all. We found out this year, sometimes we'll get producers send in several hundred samples, and many of these test negative. And what they're doing is just having a look at their field and to see what the situation is within the field itself. And these fields that aren't showing many symptoms, growers are just concerned about is blueberry scorch or shock present. That biases the results to more negative samples. On Vancouver Island and the Fraser Valley, we've recently detected blueberry virus S. It's another carlavirus that's closely related to blueberry scorch. We dug up some plants in a field that had scorch symptoms, and we pot them here at the lab, and we're using them for purification for virus antibody production. The blueberry virus S identity was confirmed using virus-specific PCR primers from Dan Villimore's group at the Arkansas Clean Plant Network. The primers for this are published in Phytopathology. A Washington isolate, WA2, was originally thought to be a blueberry scorch isolate. It is now known to be blueberry virus S. It shares 97% nucleotide sequence identity in the coat protein and 96% amino acid identity in the coat protein. So it's a strain of blueberry virus S, not scorch. The monoclonal antibody we use for routine testing here at the lab detects both blueberry scorch virus and blueberry virus S. It's fortuitous because we made this antibody back in 2008, and we field tested it with Bob Martin's group at Corvallis for 3 years in the field, compared with his standard polyclonal antiserum, and we had no disagreements in the testing result. We continue with the monoclonal antibody simply because it gave us a better signal-to-noise ratio in ELISA. We also have been testing cranberry over the years. From 2008 to 2025, we tested just over 1,600 samples from 46 locations in Canada. That's Eastern and Western Canada. Samples have been tested for scorch and shock. More recent samples have also been tested for blueberry virus S, and tobacco streak, as well as blueberry red ring spot. Incidence of scorch varied in these cranberry bogs from 0 to 100%. So same virus, blueberry scorch, can go to town on cranberry as well. What we've noticed is we see very little in the way of symptoms in cranberry versus blueberry. So when cranberries and blueberries are sometimes grown in close proximity to each other, we suggest that aphid management be carefully done. And Carolyn will talk about aphid management in a talk later in this group. To give you an idea of what blueberry scorch looks like, it's typical of carloviruses. On the right-hand side of the screen, you'll see an electron micrograph of blueberry scorch. It's a flexuous, semi-flexuous rod. Carloviruses are mostly in the 650 nanometer by 13 nanometers across. And on the left-hand side of this panel, you'll see a cesium chloride density gradient of purified virus. That's blueberry scorch, that opalescent band in the center of the tube. We extract that band, dialyze it against the phosphate buffer, and use it to inoculate mice for monoclonal antibody production and rabbits for polyclonal antibody production. The polyclonal antibodies are used as coating antibodies in ELISA to trap the virus. The monoclonal antibody is used for specificity. It only reacts to blueberry scorch, and this monoclonal we're using also detects blueberry virus S. So our serological plans for 2026 is to continue purifying blueberry virus S and scorch to produce new polyclonal antibodies. Because we do so much testing, we use a lot of antibodies, so we want to make sure that we don't run low. And we want to produce and screen a new panel of monoclonal antibodies to look for monoclonals that will distinguish between blueberry virus S and blueberry scorch virus. We have also polyclonal antibodies that have been raised to cloned luteovirus coat protein provided by Jim Madison's group at Simon Fraser University. And we've also raised monoclonal antibodies against the same cloned coat protein. And we'll be screening against several of the newly described luteoviruses. What's key here is we need type strains to do that screening. And Jim Madison's group is trying to get us the different luteoviruses for doing this final screening process. We need L, M, and N strains to see, do these antibodies detect all the strains, or do they detect specific luteoviruses? Other plans for 2026, as I say, we've introduced a direct ELISA for blueberry scorch and blueberry virus S. It gives us improved sensitivity and detection, and that may be partially why we're seeing an increase in the number of blueberry scorch positives we're seeing in 2025. We are also working with SignalChem. Ryan Yen is the man at the doctor at SignalChem doing the development of a point of care test, a sensitive point of care test that can be used for blueberry scorch and shock detection. We offer an endpoint multiplex RT-PCR that was just described by Villamore et al. In 2026. The online paper is available as well. I'll put that in the references for the written portion of this. We continue to work with Carolyn Teasdale and Dr. Lauren Erland to improve sampling for better detection of blueberry scorch and shock. And we've started the transition to look at HTS for testing early generation germplasm. That HTS, we won't be offering that as a service probably for till late into 2027, because it's going to take time to get up and running with that. And with that, I will stop. And if there is questions, I'll try to answer them for you.
Thank you, Peter. We have time for one question. So sorry. One question.
Yeah.
Maybe two. So okay, I see there is a hand. Yep.
Hi, Peter. It's Ryan from SignalChem. Thank you for giving your update. My question is it important to distinguish between BVS and blueberry scorch virus? I'm just trying to understand how closely they are related, and would they sort of have different treatment outcomes for if
The treatment would be the same in terms of they're both aphid transmitted. They both give the similar symptoms in the cultivars that I've seen here that we have potted here at the lab. There's ways of distinguishing between the two with multiplex PCR. So if you wanted, you could, but I would say the incidence of blueberry virus S, it would fall between maybe 1 to 5% in the Pacific Northwest. As when I last spoke to Bob Martin a year or so back, he said blueberry virus S would be about 3% of what they would see with things like with scorch symptoms. So it's not as big a thing, but as I say, we could probably sort it out with monoclonals, multiplex PCR, or if you wanted to do it with your point of care test, it's possible to do that too. But as I say, it's a small percentage of what we'd see in the field, and that would depend on how keen growers are to look at a blueberry virus specific test.
Thank you.
Hello.
Okay, one last. Yes, thank you.
Something that I'm curious about is we've always been taught that honeybees were the vector for shock, and aphids
Yes.
were the vector for scorch. Is that a correct reading on
Well,
what actually happens in nature?
I would think anything that visits the flowers would be capable of vectoring blueberry shock, but aphids are required for transmission of the carlovirus of blueberry scorch.
And would aphids not likely transfer shock then?
No, it's mostly pollen transmitted, and it would be the same thing for tobacco streak that you would see in eastern parts of North America. Either both Eiler virus, both transmitted in the same way, and just like scorch, they're unevenly distributed in the plant. This is our enemy, trying to sample material that has virus in it without missing the virus in symptomatic plants. And I think some of the work that we've started to do with Dr. Lauren Erland and Carolyn Teasdale will help identify the best tissue for sampling. We've noticed in our own positive control material, as you go from the tip towards the base of the stem, the virus concentration changes. The tip of the plant, the very extreme outside leaflet, generally has lower titer than as you move towards the trunk or the main part of the stem. So that's something we've noticed in our control plant. Is that in plants? I'm not sure if that is true for all cultivars, but it's maybe something we can look at with Carolyn and Lauren Erland this summer.
Thank you. Thank you, Peter, for your beautiful and very, very contextually very rich presentation. Thank you. Thank you for your time.
You're welcome.
I will also remind, if someone was not there at the beginning, there is a sign-out sheet before you leave this. Like if you want your credit for your pesticide, please make sure you sign. Please make sure you sign. So I will move on to our next presentation. Our next presentation is on new cultivar susceptibility to scorch virus. Please welcome Dr. Lauren Erland, Assistant Professor and Director of Berry Environmental Resilience Research and Innovation Center at University of Fraser Valley. Lauren.
Okay, well, it's great to be all here with you. Okay. That's a great start. Words are hard. We'll see how this goes. At least they give me a clicker that's only green and red, so I can't mess it up that badly. So today I'm going to talk to you a little bit about a project we started in 2023. This is a very collaborative project with Carolyn, as well as Jim Matson's lab at SFU, Dr. Renee Prasad at UFE Agriculture, and Siva, who has abandoned us through retirement at the ministry. So we're holding it against him. So I basically left the heavy lifting for Peter to do because I knew he was talking before me. So we all know what blueberry scorch virus is now. What I mainly want to point out is that we don't have great information on kind of virus susceptibility. We know from what Michael said yesterday that we don't have any truly resilient cultivars, but we probably have some that are more and some that are less susceptible to the virus. And so this is some work from 2000 that was done in some cultivars, but not ones that tend to be used for planting here and not any of the new cultivars that we're considering. And so the goal of this project was to evaluate some of the cultivars that are of interest in BC. As Peter said, there are lots of challenges specifically in finding blueberry scorch virus in plants, and then the long latency periods and the number of cultivars that are asymptomatic, as well as the fact that we started this in 2023, which was a low aphid year, have kind of all highlighted how challenging virus research can be. And I see some heads nodding for anyone who has tried this. So this is roughly what we did. So we started in 2023 by getting clean nursery stock of the plants we were interested in. We found a site that had high positivity rates for blueberry scorch virus. We put the plants in the field to try to get them infected. Then we brought them back to UFV, maintained them in ideally aphid-free conditions, and then have been monitoring them ever since. So these are the varieties that we included. So we have Arabella, Peachy, and Loretto Blue, as well as everyone's favorite Calypso. We have 3 new selections from the blueberry breeding program. I'm just calling them 154, 158, and 47 because there's too many numbers. And then Elliott, Blue Crop, and Duke as kind of currently planted varieties. For these, we had 20 plants of each, except for the blue crop, we only had 8, and 16 went into the field, and we held 4 back as kind of like a non-exposed control. Some notes on how that didn't work later. So this is just to show you the field that we were working in was mostly positive. So anything that's blue is a plant that was tested across the field across 4 different blocks. If it was tested positive by ELISA, by Peter and Gerda at Phytodiagnostics, it's blue, and then the green are the negatives. So we had very even virus coverage, which is great for us, and I'm the only one in the room who's excited to see a block of solid positives. This is what it actually looked like in the field. So we put plants in buckets and we tried to stick them in the place where the aphids would jump between the plants most. So we basically nestled them in the canopy. And then the poor students got to go in and hand water these through May to July. So they loved us. Whoops. Oh, I got too click happy. And then this is just to say aphid monitoring happened the whole time they were in the field to make sure aphids were present. This was led by Dr. Renee Prasad. And you can see aphids. So we had aphids even though levels were low, so we were optimistic we would get some transmission. After kind of the peak aphid period, we pulled them out of the field and brought them to UFV. And then the fun began. So after 1 year of exposure, we actually only found 3 plants out of the 200-ish that we had that were positive via ELISA. This is a picture of a scorch positive plant. Doesn't it look healthy? All of the plants we have basically are asymptomatic. So we're getting positive tests, but we're seeing very few symptoms. We know that asymptomatic plants and long latency periods are a thing, so we're not surprised by this. And as we moved into 2025, we expanded this testing to include qPCR so we could get those really early low-level infections. As Peter was mentioning, the ELISA is just a bit less sensitive. So 2025, 2 years after exposure, this is kind of the breakdown across our varieties. I've ranked from the most positives to the least positives from top to bottom. So green is positive, blue is negative. No, I don't know why I switched them. Someone should quality control my slides, I'm sorry. So at the top, I will just let you read the cultivars because I don't want to say their names out loud and get in trouble, but Calypso's at the top. And so one of the interesting things we see as we look through this is most of these positives were from qPCR testing. So the yellow bars on this version of this slide are the ones that tested positive by ELISA. Most of or all of the ones, like all of the like 2 that had symptoms were ELISA positives. Most of the positives are asymptomatic, and they were by qPCR testing. So if we look at things that are like over a 50% positivity rate, which kind of aligns with what Peter was showing before, these are our 3 that are popping up as kind of over the 50% positivity rate. So Calypso, Loretta, and Peachy. I'll say all of the plants before they went in the field tested negative. We checked them all first. So this is since the trial started. But to highlight the fact that research is not perfect, and we are, growing these plants in pots outside. They're all in a screen house, so they're under insect exclusion netting. They're outside behind our greenhouses at UFV, but they are potted plants that we're trying to keep alive, like through the winter. So we did have some deaths in these plants. So those are the purple bars, and we saw some variable deaths across cultivars, so I'm not sure what to tell us there. But the thing that I thought was really interesting is that we actually have some positives in the control plants. So the plants that we kept back at UFV, some of those actually ended up getting scorch. So my take home is when I'm trying to make things get the virus, they don't get it, and when I try to not give it to them, they all get it. So when we kind of think about revising our positive rates based on this, we can kind of lump anything that tested positive from field exposure, anything that tested positive over just the duration of the trial into all of our positive bins. We end up lumping in the blue crop in as well as over a 50% positive rate. So it looks like it's below the 50% of plants bar, but that's because there was only 8 plants total. So hopefully I haven't made that too confusing. So Calypso, Peachy, Loretto, and Blue Crop all were over 50%. And you can kind of see the breakdown in the numbers there. I feel like Eric's gonna call me a nerd because I have put too many numbers, but that's okay, we'll talk to him later. The good news is some of them had very, very low positivity rates. 47 and 158 out of the BC Blueberry breeding program, very low, as well as Duke. So I think that this is all kind of informative. The main thing I want to highlight in this though is these are all asymptomatic. So like there's probably, I'm going to say something really unpopular and then I'm going to run away really fast, but there's probably more blueberry scorch virus around than we maybe realize, but it also is maybe not as big of a deal as we kind of initially had the feeling of, because there's a lot of just asymptomatic plants probably sitting around that it's not causing you problems. So I'm just gonna throw that out there and then I will hide behind this podium. So overall, where are we going next? As Peter mentioned, we're gonna work on trying to get some more information on like sampling in locations in those plants. So we know that if we take 10 leaves off of a single plant, 50% of them test positive in a symptomatic plant. So it is hit or miss on catching where the virus is. We're going to continue monitoring for symptoms and see if those rates change, both in terms of asymptomatic versus symptomatic plants, but also more qPCR and ELISA testing this year. And we'll just keep you guys posted on what we learn on that front. So with that, I'm going to say thank you to the long list of students who have been taking care of these plants diligently, all of our collaborators on this, and I think we've got 5 minutes for questions. Please be nice to me.
I think something they'd like to get cleared up is the asymptomatic plants are still carriers of the virus. So if your field is 25% asymptomatic and you don't have any losses in your crop, they're still carriers, so that the rest of the farm could all be asymptomatic in 10 years if you had any kind of vectors that were still active, which is usually the case. So at some point then, are we to assume that some varieties are capable of just being normal, in quotes, production units with the asymptomatic, or what would you conclude by that percentage of asymptomatic plants?
Yes, okay, so full disclosure, I'm a researcher and I'm not a blueberry grower, so you all probably know better than me from a production context, but I would say that yes, we probably have some varieties, and we know blue crop already is one of them just from what we've seen in the field, that will just probably mostly stay asymptomatic. So like, you're not going to see production impacts. The thing that seems to be happening, and I don't have like solid data to back this up, but seems to be happening is like when you have like a year where you have other stress factors, if the plants are positive, it's going to make it worse in that year. But my inclination, this is a lot of hedging, is to say that like even though you've got some symptomatic or some asymptomatic plants in there, like it's not affecting you year over, like every single year, because there do seem to be some varieties that you just have really high asymptomatic levels on, but we probably need more information on that for me to be able to say anything definitively.
But they can go from asymptomatic to symptomatic sort of randomly in your mind, or is there a stress factor that converts them?
I don't have enough data to give you an answer to that. I would love to get more information on that because I think it's a really good question.
Okay, thanks very much.
Hey, Lauren, it's Eric back here. Great presentation. Thanks so much for that. Excellent to see this work's getting done. I just had a question about during the exposure period in the field, did you notice any difference between the cultivars in the amount of vegetative vigor, new vigorous growth on those baby plants?
Ooh, good question. The plants were more or less of similar size when they went in the field, and they all went in looking healthy. However, they were like the nursery, like the ones we got from commercial nurseries were a little bit bigger. So the Elliott and the blue crop were a little bit bigger, but they were more or less similar going in. I think we saw strong differences in health of the plants over the course of the trial, which is probably due to management and care of the plants. So drought stress, heat stress, soil pH stress, like various and assorted things that make it hard to keep plants alive in pots. Yeah.
Thank you.
Hi, Lauren.
Hey.
Jen from ES Crop Consults. I'm just curious, maybe you showed this. You said you monitored aphids, but did you monitor the levels for each variety? So can we conclude those varieties that had over 50% infection, was it because they just had more aphids on them?
So I would refer for specific aphid details probably to Renee because she did the monitoring, but I know that she found both winged and wingless aphids present across all of the varieties.
Thank you, Lauren. Great info. But just a little point, I think, with the asymptomatic plants. I mean, we've been farming in Surrey for 46, 47 years. A lot of scorch out there. I think one of the biggest things is just the way the plants look. So say blue crop and Duke, which can be asymptomatic, but their production will still drop tremendously. And the plants such as Elliott and Calypso, we're finding when scorch gets in there, they look dramatically weak and dried out very quickly. But the blue crop, the asymptomatic blue crop, still will transmit the virus tremendously, especially if aphids aren't being controlled. So if there are fields out there that are, and there's lots of these older fields that have a huge amount of scorch, and if they're not pulling out plants and they're not controlling aphids, I mean it's an exponential growth as you know firsthand, and those aphids will travel miles, right? So I mean, it's these newer plantings and especially the newer varieties such as Calypso being planted, and if there's fields within miles of them of scorch-infested asymptomatic fields, I mean, it's not long that those fields will be infected also, right? So I think I just want to clarify with the production, at least from my experience, that there will be a dramatic drop-off, even if the plants look good, and they'll look good even right through to blossom. It's just once they get into fruit set, they just won't pollinate, won't fruit set, and they'll have a fraction of the fruit, and depending how old the plant is, it could be 10 to 15% of what their potential is for some of those fields.
Yeah, so I guess my time is almost up, but I will say quickly, it might be a question of how you define asymptomatic, because I think if you're starting to see those losses, it's not asymptomatic anymore. So, and then the other thing I would say is yes, I mean, best practice is to not have any virus reservoir out there. Like you want to control your aphids. You don't want to have like a virus spreading party of blueberries. I mean, but also like I don't want the message from this to be like Calypso seems to have like the highest positivity rate. Panic. Like I think like some caution, I guess, in the messaging here, and we need more information. Basically, blueberry scorch virus is very hard to study and very complicated, and the more information we can get, the better. But yes, to all of that. So thank you very much.
It was interesting, isn't it? To understand different varieties and all. Thank you. Thank you, Lauren. Our final presentation before the refreshment break is on blueberry scorch virus spread in Calypso field. And please welcome Carolyn Tisdale, industry specialist for berries with the BC Ministry of Agriculture and Food. Carolyn.
Thanks, Sudeshna. Okay, I'm going to present on our 4th year of field research looking at the rate of blueberry scorch virus spread in commercial fields. And this research was done by Patricia Gendron, who I believe is in the back, just raising her hand. I don't want to take credit for all this work. The research was supported by the BC Ministry of Agriculture and Food, and it was designed by the ministry. And research funding was provided by the governments of Canada and British Columbia through the Sustainable Canadian Agricultural Partnership, a federal, provincial, territorial initiative. I'll start with a disclaimer. Opinions expressed in the document are those of the author and not necessarily those of the Government of Canada and British Columbia. So the objective of our research was to monitor the change in blueberry scorch virus infection over time in fields under good management in British Columbia. And the reason we wanted to do this was because back in 2021, we saw a lot of virus symptoms in blueberry fields across the Lower Mainland. And there was concern that this virus was spreading faster than before. However, we did not have research to show this, and so we wanted to look at it more closely. When blueberry scorch virus spread was last studied in BC, the percent increase in diseased plants was from 2 to 5% per year. That was back in 2001 to 2004, research done by Lisa Wagner and Mark Sweeney. And what they found was that it was closer to 2% when aphids were controlled and more like 4 to 5% when aphids were not. That was done, I believe most of the fields were in Richmond, and they were blue crop and perhaps rubel. They were older varieties, so they weren't the same varieties that we're looking at now. In this research, we monitored the extent of virus spread in 5 blueberry fields. I'm just going to present the results from this last year. So we previously had 7 fields. We lost 2 of the fields this year due to the growers deciding not to participate. But we still had 3 Calypso fields, 2 Duke fields. All of these fields we started monitoring in 2022, and we selected them because they had low levels of virus symptoms. So we were hearing concerns from growers that, okay, if I plant a new field or I have a clean field but my neighbors have scorch, can I actually keep it out of my field? And so that was kind of the question we were trying to answer. In each of these fields, we selected 200 bushes, and they were situated in 4 plots across the field. So I just want to draw your attention to that diagram because I'm going to show it repeatedly throughout the presentation. Think of that as a top-down view of the field with each little white square indicating a plant. It's very simplified. And the green ones are the ones that we sampled and monitored every year over time. So they're divided 4 plots. Each plot has 5 rows times 10 consecutive bushes. So really important to note, the exact same plants are being tracked over time. We monitored these plants annually for symptoms in June and sampled them for shock and scorch viruses in the summer. All virus samples were sent to phytodiagnostics, so they were tested using ELISA. Most of the fields were monitored for aphids bi-weekly in 2023, 2024, and 2025. And the growers were surveyed at the end of each growing season about their management practices and their yields. I'm going to jump right to the results for the Calypso fields. The good news, scorch virus is spreading slowly in young Calypso fields where aphids are being managed. So the percent increase in scorch-infected plants ranged from 0 to 1% per year, or 1 to 1.5% over 3 years. This is, if you recall, I said back in 2001 to 2004, it was 2 to 5% per year that was being found in that research study. So here we're finding a lower rate of spread. This may be expected because the fields are pretty clean. There's not a lot of inoculum in the fields themselves. All Calypso plants that tested positive for scorch virus were asymptomatic. This is also not surprising given that these are younger fields and new infections, and we know that there can be a 1 to 2 or sometimes even 3-year latency period between the time of infection and the time of symptoms. I'm going to go through each of these fields in a little more detail. So this is the first Calypso field. It was planted in 2018. It had a neighboring blueberry field to the west. It was receiving 5 to 7 aphid sprays annually. And the peak aphid levels in terms of the percentage of shoot tips infested with aphids are shown there. So 5% in 2023, higher in 2024. We did have that extended bloom period in 2024 and the aphids creeped up at the end of bloom. And then 7% in 2025. And those are the yields reported by the grower. So here are the results for that one field, Calypso 1. On this slide and on following slides, what you'll see is I've given the same diagram over time. So it's the same field with the results in 2022, 2023, 2024, and 2025. Again, the green squares are the plants that we sampled. Those squares remain green if the plant remains healthy or tests negative for scorch virus. If it tests positive, the square turns color. And so you can see in this field, we did not have any scorch positive plants until 2025 when we had 2, and they're kind of in the middle of the field there. They've turned blue. And neither of them were symptomatic. Neither of them were removed. Calypso 2 was planted in 2020. It had neighboring blueberries on all sides. It was receiving 4 to 6 aphid sprays annually. It had low aphids in 2023. This field, we do not have aphid data in 2024 or 2025 at the request of the grower. And there are the yields reported by the grower. Here are the results for Calypso 2. So in this field, we had our first scorch virus detection in 2024 on the north edge of the field. And then we had 2 additional plants test positive in 2025. The plant in 2024 was removed, so we don't know if it would have developed symptoms. The 2 plants detected in 2025 did not have symptoms at the time of detection. And Calypso 3, so this field was planted in 2021. It had neighboring blueberries to the northeast and the west. It was receiving 2 to 3 aphid sprays annually, and its peak aphid levels are listed there. So fewer sprays, but the aphid levels are very low because the sprays were well timed. And there are the yields reported by the grower. So this field, we were surprised. We detected 4 scorch positive plants in the first year of the study, and we've had 1 additional scorch positive every year since. You can see that initially the plants were kind of scattered throughout the field, but in recent years, most of the infections have been in the southwest. I suspect this is because of the wind direction and the neighboring fields, and aphids are likely blowing in, and that's perhaps where they're landing. This field, initially when we had 4 plants test positive, 3 of them were removed, but 1 of them was left in. So we've been able to look at that field to see if it develops, or that plant to see if it develops symptoms over time, as well as the one detected in 2023. And interestingly, those 2 plants still are not showing symptoms. So it's making us question whether the latency period is perhaps longer than 1 to 2 years or 3 years. I think the coming years will tell. This slide is just a summary showing the percentage of plants with scorch in 2022, 2023, 2024, and 2025 in each of those 3 fields. So you can see that the percentage of plants infected remains very low. I think this might be more interesting. This is the percent increase year over year, and then the total increase. So over that 3-year period, the total increase was 1 to 1.5%, depending on the field. Now I'll shift gears and talk about the mature fields. So we had 2 mature Duke fields where we were monitoring rate of virus spread. In these fields, the rate of spread really varied depending on the initial extent of the virus. So the percent increase ranged from 0.5 to 1.5% or 3% over 3 years in a mature Duke field that started with low levels of virus. But we did have one field that actually had high levels of virus, and in that field, the percent increase ranged from 3.5 to 10.2% per year, or 18.8% over 3 years. And that, as I said, that field started with high levels of virus. 2 other really important results, I think. Scorch virus symptom expression varied by year, and many infected plants did not show symptoms. These are more mature fields, so we assume that some of the plants were infected before, like more than a year or 2 before we started. I'll go into the fields in more detail. Duke 1 was planted in 2008 to 2009, so it's over 25 years old. It was receiving 3 to 4 aphid sprays annually. There's its aphid levels. You can see a bit higher levels in 2024 and 2025, and those are the yields reported by the grower. In this field, you can see we initially detected 5 scorch-positive plants, and then we've had an increase over time. I wanted to draw your eye to that blue line there with the percentage of the scorch-infected plants that were symptomatic each year. And you can see that it was never more than 36% of the plants. Again, we don't know how many of them are new infections. I mean, we can tell from the annual results, but in 2022, we don't know how recently those were infected. But at that time, only 20% were showing symptoms. And here's Duke 2. This one was planted in 1995, so it's now 30 years old. It had neighboring blueberries to the west, east, and south, 2 to 3 aphid sprays annually, and there are its peak aphid levels. And those are the yields reported by the grower. So I want you to notice that this field is still producing. If you've seen these presentations in previous years, this won't be a surprise, but this is the field where we thought that we were starting with a clean field that did not have many infections. But when we tested the plants in that first year, 113 out of 200 of them tested positive for scorch virus. However, only 4 were symptomatic. We have seen low levels of symptoms over time, with the exception of 2024, which was a year when scorch virus was really apparent across the Fraser Valley. And that peaked at 40% of the scorch-infected plants showing symptoms. Here's a summary slide for those 2 fields, showing the percentage of plants infected with scorch over time. And then the percent increase year over year. And you can see the total increase from 2022 to 2025 really varied between those 2 fields. So to summarize, the rate of scorch virus spread can be kept low when fields start with few infections and aphids are managed. Symptom expression varies by year. Less than 50% of the plants in this study that were infected with scorch showed symptoms in any year. And aphid management is likely the most practical scorch virus management strategy because it is very time-consuming and costly to test all of your plants if they're not showing symptoms. And from a business decision, I don't know that it makes sense to remove infected plants when they're still producing. However, there is still some really important pieces of information that we do not have, and some of them were raised in the previous questions. We need to know how long it takes for infected scorch-infected Duke and Calypso to die. We need to know what the impact is on yields over time. When I talk about the symptoms that we were observing in this research, we were looking for blighted flowers, blighted leaves. Those are the key kind of classic scorch virus symptoms, but there may be a yield impact that was less obvious. We want to know what the impact is of mixed viral infections. And we also, I think, need to know what percentage of asymptomatic scorch-infected plants are going to develop symptoms over time so that we know whether, this is just a management decision that we're going to have to sort of factor in, that it'll be a longer time before they develop symptoms, or if they never will. Thank you. I won't go through all the names on the slide, but a big thank you to Trish Chandron from Coast IPM and our grower cooperators for giving us access to the fields. And I think that I've used up most of the time, but I may have time for one quick question.
Hey, Carolyn, it's Eric in the back over here. Thanks for the awesome presentation. I just had two quick little questions. You made a comment about Calypso 3 about how one of the plants had been removed. Could you comment about whether or not other plants that had tested positive had been removed over the course of the study? And then could you also make a comment about whether you think with these heavily infested fields, whether there's a tipping point and that those fields begin to snowball compared to the fields that have low levels of initial infection?
So your first question was whether plants had been removed. With the Calypso fields, it was mixed. So I would say, I mean, we could go back through the slides, but I'd say about half of the infected plants were removed. None of them were symptomatic, though. So we do have a few that we can continue to follow over time. I think this is the last year of this research project in that we don't have any more funding for the whole project, but I can hopefully go back and track some of those individual plants for a little bit longer. The second question was about whether it will snowball. And do you mean by percentage of plants infected in the field? Like the infection, the rate of spread is greater once you get to a certain level?
Exactly, and that
Yeah.
continued aphid management will be less effective in stopping just a progressive decline.
I mean, I do think that when you get to a certain level of infection, aphids are more likely to be landing on infected plants than uninfected plants, so they're more likely to be carrying the virus, and wherever they go, whatever's remaining, they're going to continue to infect. But I think, you know, there's certain cultivars where if you get to that level of infection, you'd probably be seeing a lot of dieback. You know, any of you who have had Elliott, we saw a lot of dieback with Elliott. So I think I don't know if we're going to see decline in the field over time. I think time will tell, and it does depend on stresses. But I do think that there is an increased rate of spread once you get to a certain level.
Carolyn, just a question regarding if you guys are collecting data or keeping information on the level of farming on these neighboring fields, because that might play a little bit into the research, because certainly in many regions there's some fields that are still farmed pretty aggressively and sprayed, and then there's others that are almost vacant and just even just let on the side and no aphid control. Are you presently in this research taking that into account, or moving forward? Is that something to plan? Because I think that there'd be value in that.
Yeah, absolutely. It would be important to know whether aphids are being controlled in the neighboring fields. We don't have that, so we didn't talk to the neighbors. I can say that most of the fields had scorch in them that were nearby, but aphid control, we didn't get to that extent of surveying all the surrounding fields, and that would be useful to know. I think we are out of time, so wrap it up there, but I will be around if anyone has any further questions about this research.
Thank you, Carolyn. It was very, very useful presentation. Please let us know what you thought about your experience in this conference. By end of this conference, we would love to hear your feedback on the various sessions you attended and the conference experiences you had. To do so, you can either scan the QR code appearing in the screen right now, or if you have downloaded the event app, you can access the form at any time by clicking on the survey button appearing on the home screen or program page. Those who submit their survey response by Thursday, January 29th, will be entered into a draw to win 2 complimentary passes to the 2027 Lower Mainland Horticulture Conference. We would really appreciate your feedback, so please do that. If you are earning pesticide recertification credit, again, just for us, if you can please see one of our volunteers at the back of the room and sign the session's attendance sheet. With that, we will now take a refreshment break, and we will get started again at 11 a.m. I think it's past 11 a.m. So we will give around 15 minutes, maybe 11:10 sharp. Enjoy your break. Thank you. Thank you very much.
Hello everyone, we are in day 3 at BC Blueberry Council and Pacific Agriculture Show 2026. Here with us, Rick, who is the ex-executive director. Hello Rick, how are you doing?
Oh, wonderful. Great show here.
Are you enjoying these 3 days?
Oh, absolutely love it. And I'm so impressed with the BC Blueberry presentation at this forum. It's just simply amazing.
Let's start with your journey. How you came in touch with the BC Blueberries, and how was the journey with BC Blueberries?
So this is the second time I did some work with the BC Blueberries as a consultant. Back in 2018, worked with a great group of people and leadership for the council. Totally different organization than what I see now. Which is very, very positive. And then last April, I had a call to see if they were going through some transition, and asked me if I would step in to run the organization and help facilitate the hire of the new executive director, which I took on with pleasure.
That's great. And you have seen BC Blueberry Council from a very close look. You met with the farmers, growers. What have you seen from the past years till date?
Well, I also had the pleasure towards the end of my term this year with the BC Blueberries was to work closely with Sudesh, the new executive director. And so we had a lot of conversations, and I've just been blown away with all the transitions and transformations she's been making, and her staff with the Blueberry Council. The relationship with the growers and the packers, the communication, the transparency, the leadership with the board as well. Yeah. It's all been extremely positive.
That's great. And these 3 days are going very well. If we just talk about day 3, which is today, weekend, people are coming here, joining BC Blueberries, having a talk with all the team of the BC Blueberry Council. That's a great one, because this is the way how we spread knowledge about the BC Blueberries.
Oh, yeah, exactly. It's a very strong presence. It's getting out in the community, letting the community know that the council's here. It's also engaging with the members of the community, but also an opportunity to expose the farmers, the growers, and I'm sure there's some packers and receivers here as well. So it's really waving the BC Blueberry Council flag.
What's the expectations from BC Blueberry Council in future, and what's the expectation from the farmer or the growers in basically the field of blueberries?
Well, I'd like to say that I know, but I don't know totally because I've been away now from the council for the last 3 months. However, what you are seeing is even the promotion of the council at the booth, right? The presence, the online presence with the BC Blueberries, the level of and quality and professionalism of the presentations is very, very, very high. But it's really engaging with the blueberry ecosystem, right? With all the members and the stakeholders, and that's so positive. And from what I can see too, is advancing the government relations side of things, engaging with the government and different collaborators from that point of view. So it's not a passive involvement in what's happening here in British Columbia. It's a very active and aggressive. And I can also see what's happening internationally with new partners that are happening within the council, exploring different countries that they haven't explored before. And again, with Sudesh's experience and background with international agriculture, it's going to benefit BC Blueberries.
That's true. And what are your views about these kind of events? If you just talk about Pacific Agriculture Show 2026, a lot of people are visiting this show, a lot of manufacturers, company owners. This is a great thing if we need some advancement in technology, even moving forward in today's year.
Oh, definitely. And it's amazing. So when you look around, you see all the different partners within the agricultural system, whether it's finance, whether it's equipment, whether it's growers. Spent some time talking to the research community, the academic community. To our right is University of the Fraser Valley, and Simon Fraser University is here as well, the research people. And so these are all potential collaborators and partners for the BC Blueberry Council as well. So this is a huge win-win, and whenever they have an opportunity to participate in an event like this.
Last but not the least, what would be the message today for all the viewers watching us?
Oh, just watch this organization take off and grow even more. It's just simply amazing with their leadership, very supportive board of directors, and I, having observed the town hall in Abbotsford earlier this year, I think the level of engagement going forward is going to be very, very positive.
Even the town halls were all the 3 town halls were very successful, and now these coming monthly meetings are really the crucial role in BC Blueberries.
So, and you were asking earlier about some of the changes and transformation. That level of engagement, I never personally saw that in the past, and I think it's just so helpful to the council to do this. And so the growers and the packers get to know the council and the leadership and the programs, and likewise the council get to know them as well.
Thank you so much, Rick, for sharing all this information and your views about the show, about BC Blueberries and the transition which happened recently. Thank you so much for your time and talking with Sanjha TV. It's nice meeting you.
My pleasure. Thank you very much. Okay,
Thank
take
you.
care.
Hello, everyone. It's day 3 here with us, Sanjha Gill, who is the chairperson in BC Blueberry Council. Hello, Amrit. How are you doing?
Good. How are you?
Good. Nice to see you here.
Yeah, thank you. Good to be here.
Yeah. So, what are your views about the show and BC Blueberry Council?
The show is great. It's great to get out here, meet growers, meet other industry professionals, and network and get the BC Blueberry out there and get as a brand.
Definitely. And these 3 days were the successful ones. If I just tell you that the growers are coming, farmers are coming, meeting you guys, your team, and it's a great one for spreading all the education and everything.
Yes, exactly. That's the whole point. Was want to get the BC Blueberries out as a brand and make sure the farmers have they know that we're here to support them, and they can come talk to us.
What are the new things upcoming from the BC Blueberries team?
We're rolling out some new initiatives. We're mainly focusing on a lot of marketing now, so focusing a lot within Canada and doing rebranding. Like, we have a brand new logo, we're rolling out a new website. So just making sure we have a lot more brand awareness and people know that we're out there. So we're here to support the farmers and also to better the industry and make sure that the industry has growth.
Definitely. And after the 3 successful town hall meetings, people are loving what are you doing, and these monthly meetings are the crucial ones, and people are just waiting for these monthly meetings.
Exactly. Yeah, the town hall were a success. We got a lot of information back. So we think it's important to get feedback from our growers so we know exactly what they want. Because at the end of the day, we're here to make sure that our growers feel supported. So the town halls were a success, and we're going to start rolling out our new education and monthly grower meetings, and so that we can hear more of what they have to say and offer them education and whatever support they need.
I love your t-shirts. How can we get these ones?
We're actually getting some t-shirts out right now, so if you like some, we can give you some after the interview.
Your team is fully ready to meet people, and it feels like the BC Blueberry team is ready for rocking everything.
Yeah, 100%. Where we got a lot of new blood, and we're going in a good direction. So we have a lot of momentum, and we're looking forward to this new year, and we're looking forward to just bettering the industry and bettering the lives of the farmers.
Last but not the least, what would be your message to all the viewers today?
Buy BC Blueberries, buy local, and support BC Blueberry farmers.
Thank you, Amrit, for sharing your words, views, and your lot of information about the BC Blueberries. It's nice talking to you.
Yeah.
Thank you so much.
Thank you for having me.
Thank you.
Yeah.
We are having 2 strong pillars of BC Blueberry Council, Sudeshna and Sunny. How are you doing, guys?
I'm very good, thank you.
I'm great too. I'm enjoying this show a lot.
This is today's the final day, and what do you want to say to the viewers today? Because all the 3 days, I want a complete message from you guys about these 3 days.
Sure. So last 3 days, it was my first FedEx show, and I never been in any Pacific Ag show before. Of course, not my first rodeo, but the first Pacific Ag show. For me, what I felt is lots and lots of engaged grower, engaged footfall who wanted to know more about many things. And that surprised me because normally I get people who comes booth to booth, but we got lots of questions, lots of discussion, and good discussion and useful discussion. That made me feel very good about this show.
And what about you, Sunny? How you feel about these 3 days?
It was amazing because I went to these meetings, trade show events before, and the discussion was very short and quick. And the type of discussion we're doing now is very deep, very insightful, because we have a lot of these programs that we need to explain to a lot of people. Take time to explain so they can explain to other people.
Yeah.
Right. So it's very interesting. They're very interested, so they have other questions. So the average conversation is lasting at least double, triple time. Like, I remember you guys were trying to take a picture,
Yeah.
but those guys were so interested in what we had to say that we were so engaged. And there's like that all day long.
I'm very happy to see your team growing and being more stronger day by day, and that's the most important one, working collectively. Yep. And what would be your message for the upcoming things to all the viewers and people who are just watching us? Is something new coming from you guys?
So that's a great question. There are many new things. As we talked in our all our town hall meetings, we are coming up with Growers Clinic, we are coming up with Growers Day, we are coming, as you see, there is new logo which is launched, new website coming. There are many new programs we are going to launch. I'm also very excited that there will be upcoming new BC blueberry varieties coming. We cannot talk about it due to IP law reason, but for us it is really exciting time, and I am happy to say that watch out, BC Blueberry, what is coming. Definitely. So next 5-6 months will be very big for us, and you will see what we will do. So one by one.
What you want to say, Sunny, on this one?
She pretty much covered it, but I just want to add that a different type of communication we're going to have, trade show format. We have the monthly grower meeting, which will be a step on from this trade show. Everybody who's interested to be involved. There are different emails, the social media. There's a lot more. You could see the growers. Accounts are growing, number of followings. So I just want to mention that the communication is very good.
Thank you so much, both of you, and we would wish you all the very best for the future, and we are glad to join you every time. Thank you so much for your time, guys.
Thank you. Thank you very much.
This is all about day 3 at BC Blueberry Council and Pacific Agriculture Show 2026. ਅੱਜ ਜਾਣਕਾਰੀਆਂ ਦਿੱਤੀਆਂ ਤੁਹਾਨੂੰ ਕੀ ਚੇਂਜਸ ਆਏ, ਕੀ ਆਉਣ ਵਾਲੇ ਸਮੇਂ ਦੇ ਵਿੱਚ ਚੇਂਜਸ ਲਿਆਂਦੇ ਜਾਣਗੇ ਤੇ ਕਿਵੇਂ ਰਲ ਮਿਲ ਕੇ BC Blueberry Council ਦੀ ਟੀਮ ਕੰਮ ਕਰਦੀ ਆ। ਉਮੀਦ ਕਰਦੀ ਹਾਂ ਕਿ ਤੁਹਾਨੂੰ ਇਹਨਾਂ ਦਾ ਕੰਮ ਚੰਗਾ ਲੱਗਦਾ ਹੋਊਗਾ ਤੇ ਇਸ ਦੇ ਨਾਲ 3 ਦਿਨ ਦੀ ਕਵਰੇਜ ਅੱਜ ਅਸੀਂ ਤੁਹਾਡੇ ਨਾਲ ਸਾਂਝੀ ਕੀਤੀ ਤੇ ਉਮੀਦ ਕਰਦੀ ਹਾਂ ਕਿ ਆਉਣ ਵਾਲੇ ਸਮੇਂ ਦੇ ਵਿੱਚ ਅਸੀਂ ਇਦਾਂ ਦੇ ਪ੍ਰੋਗਰਾਮ ਸਾਡੇ ਤੱਕ ਲੈ ਕੇ ਆਉਂਦੇ ਰਹੀਏ। ਸਾਂਝਾ TV ਦੇਖਣ ਲਈ ਤੁਹਾਡਾ ਬਹੁਤ-ਬਹੁਤ ਧੰਨਵਾਦ। ਮੈਂ ਤੁਹਾਡੀ ਆਪਣੀ ਸਪਨੀਤ ਕੌਰ।